Very good. And then Jessy, you can go for it.
Okay. Thank you, Steph. Hi, everyone. Thanks so much for having me to give the seminar, and thanks so much for being here to watch it. My name's Jessy Patterson. I'm a PhD student at the University of Georgia in the US. All of my dissertation work is occurring within Ongava and Etosha. So today I'll be talking about one of my dissertation chapters, which focuses on determining the effects of artificial lighting on mammal behavior at waterholes. My co-authors and co-conspirators for this project are Dr. Steph Perriquet from Ongava, and then Dr. Jim Beasley from the University of Georgia, who are both on this Zoom, luckily. So just a quick outline of what I'll go through today. First, I want to give you a look into my entire dissertation and the other projects that the three of us are working on. Then I'll go through the project background, research questions, hypotheses, methods, and hopeful future directions. And then I'll have time for questions at the end. So the overarching goal of my dissertation is to understand the impacts of human presence on African mammal ecology and carnivore diets in protected areas. This chapter I'm focusing on today is to quantify the effects of artificial lighting on mammal behavior at waterholes, but I'm also looking at the effects of human observers on mammal temporal activity and visit frequency at waterholes. I'm evaluating the impact of tourist hotspots in Etosha on the movement of lions and spotted hyenas. And then I'm going to compare the diets of carnivores within and around Etosha. So for some background on this topic in particular, we know that wildlife-based tourism generates income and provides local employment. It also increases tourist education and it promotes conservation for animal species. It's one of the main economic drivers in some parts of the world, but especially in regions of sub-Saharan Africa, like Namibia. Several studies have looked at wildlife-based tourism in Africa, but its effects on wildlife behavior and movement are pretty poorly understood. We know there are positive impacts on wildlife. Aside from establishing protected areas and those local economic drivers I mentioned, wildlife-based tourism can also raise public awareness and education. It can facilitate conservation efforts. It promotes research opportunities. And also artificial waterholes designed for tourist access can provide vital resources for animals, especially during the dry season in arid environments. And finally, even tourists on safari can access remote regions of protected areas where poaching can occur. And so tourism can provide a protective measure to deter poachers and also look for injured animals. With that said, we also have some literature that shows tourism can have negative impacts on wildlife, including habituation to humans, reduced reproductive fitness, modifying their activity patterns and predator-prey interactions, and also increasing stress hormone production. So for example, one study showed observed behavioral changes in African elephants in response to tourism presence. They noted increased aggression, moving away from tourist areas, and increased vigilance at waterholes. We know that tourism and artificial lighting are increasing in many forms worldwide. However, most of the studies that we've seen are looking at the effects of lighting on migratory birds and bat species. A few years ago, a meta-analysis showed that mammals are increasing their nocturnality by 20% in response to human disturbance, including lighted areas. So large mammals in particular had a stronger response to avoid humans. Waterholes in some protected lands in Africa are often artificially lit at night for tourism purposes, especially because many of those charismatic species are nocturnal. So many studies have been conducted on wildlife during waterhole visits, but none have actually looked at the effects of artificial lighting on wildlife behaviors and temporal activity. There have also been very few efforts to evaluate wildlife responses under varying light conditions, which is something we hope to look at. So I'll talk about that a little bit later. So regardless, my goal for my dissertation is to help develop optimized protocols that will maximize the tourism experience while hopefully reducing human impacts on wildlife. So for this project, the research questions that we're focusing on are: how does artificial lighting affect mammal behavior at waterholes? How does it affect temporal activity, the duration of visits, and the number of visits at waterholes? How long does it take for each species to habituate to artificial lighting? And how do these differ between nocturnal and diurnal mammal species? For our hypotheses, we are saying that the number of vigilance events will increase while the number of events doing other behaviors like drinking and resting will decrease for nocturnal species at artificially lit waterholes compared to the non-lit. The number and duration of visits for nocturnal species will be higher at non-lit waterholes compared to lit. Oops, sorry guys. Diurnal species will visit artificially lit waterholes more frequently at night compared to unlit, and predator species, mainly carnivores, will become habituated to light sooner than prey species or herbivores. So our entire study is taking place on the Ongava Game Reserve. We are using 7 of Ongava's waterholes for this study, which you see on the map. We have 2 experimentally lit, which are indicated with the light bulbs. And experimentally lit means that they've previously been dark, but we are installing lights and lighting them up specifically for this experiment. So that, for those of you familiar with Ongava, that would be Sunup and Pseudocris waterholes. Then we have 2 controls that have been dark and will remain dark. So those are the ones with the light bulb with the X through them. And those are Rainers and Ongava. And then we have 3 lodges, which include the Tinted Camp, Ongava Lodge, and Anderson's. So those have been lit for a long period of time. And that's another group that we're comparing. So our experiment officially begins at the end of this month and continues to December. And that allows us to encompass one season and not have seasonality effects. The lights will be mounted and cameras will be running officially by August 1st, but no lights will be turned on. So that allows us to collect baseline data without actually turning on any lights. And then by the start of September, we'll turn the lights on and collect our experimental data for about 3 months. We've installed solar panels at Sunup and Pseudocris, our 2 waterholes that we plan to light up. The solar panels are powering 30-watt LED lights, which are similar to current lights that are used at lodges. Each waterhole has an existing web security camera, which records videos continuously pending network connectivity. In addition, we're deploying camera traps at each waterhole to ensure that if we do lose connectivity, then we still have camera image data for the project. We'll also be able to use the camera image data to determine dial activity patterns, frequency of visits, and time spent at each waterhole for all species. To analyze all of the video data, we're using ORC, Behavioral Observation Research Interactive Software. It's an electronic ethogram software that allows us to play the videos and classify behaviors for each species. The behaviors we focus on include vigilance, drinking, resting, moving to, around, or away from the waterhole, and fleeing. We document the number of individuals in the group and if there are other species present when a new animal arrives at the waterhole. We're also able to time the animal's approach to determine if it takes them longer to approach a waterhole based on the lighting. For the image data, we're using TrapTagger, which is an AI software that helps process camera trap data quicker than the traditional manual method. For data analysis, for each species individually, I'm going to compare between waterhole types. So again, the waterhole types are long-term lit, dark, and then recently lit by us. I'm going to look at time spent at all waterholes, time spent approaching waterholes, the number of visits at waterholes, the number of behavior events at waterholes, and then finally dial activity patterns. So for dial activity patterns, that actually gives us a coefficient of overlap, and it allows us to visualize patterns for each species between waterhole type. So if you're not familiar with that, it creates, we're able to create figures like this, which shows us overlap patterns for each waterhole type, for example. So this is actually a figure for another project, which I think I'll be presenting in a couple of months, but this still gives you the same idea of what we'll be looking at. Each color and line type correlates with a waterhole, for example. And so if you're looking at the solid line with the pink color, that might be one waterhole type, and it would tell us that jackals at that waterhole are going to be slightly crepuscular, but mostly diurnal, compared to the other two waterholes, which would be in the green color and the blue color, where they appear to be less diurnal and more crepuscular. It gives us an overlap value comparing each waterhole to the other types, and then we're able to see if there's significant differences in their patterns of activity between each waterhole type. Based on our results from this year, we'll decide if it's useful or necessary to repeat our same experiment next year. Once that's decided and all of our data is collected, we'll then determine if we want to apply colored light filters and test for the same effects. For example, red lights are supposed to be less disruptive than white lights for animal activity. We're also interested in putting timers on the lights to turn them off early, perhaps around midnight or 1 a.m. to see if that modifies behaviors or temporal activity any differently. And with that said, I'll take any questions.